The objectives of this video module. By the time you have finished reviewing this video module, you should be able to outline the steps involved in the sliding filament mechanism of muscle contraction and describe how muscle action potentials arise at the neuromuscular junction. But before we begin, we will need to review the connective tissue components of skeletal muscle as well as its microscopic structures. When looking at skeletal muscle tissue, you will notice that there are 3 layers of connective tissue that protect and strengthen the skeletal muscle. These include the epimysium, perimysium, and endomysium. The epimysium is the outer layer that is found surrounding the entire muscle. It is composed of dense, irregular connective tissue. The perimysium is in the inner layer, and like the epimysium, it too is composed of dense, irregular connective tissue. This connective tissue is found in a skeletal muscle surrounding groupings of 10 to 100 muscle fibers, arranging them in bundles termed fascicles. The endomysium is found surrounding individual muscle fibers found within these fascicles, and it is made of mostly reticular fibers. The cells making up skeletal muscle tissue are termed fibers. They are typically much longer in length than either smooth muscle cells or cardiac muscle cells, and have more than one nucleus, which can be found just beneath the sarcolemma plasma membrane surrounding the muscle fiber. These nuclei have lost their ability to undergo cell division, so the number of skeletal fibers you are born with will last your lifetime. T-tubules are the tiny openings found all along the sarcolemma filled with interstitial fluid. These openings provide a tunnel in from the sarcolemma towards the center of each muscle fiber. The cytoplasm of a muscle fiber is referred to as sarcoplasm. In skeletal muscle fibers, the sarcoplasm contains a substantial amount of glycogen, which can be used for ATP synthesis, as well as myoglobin, which binds oxygen molecules that diffuse into muscle fibers from the interstitial fluid. The oxygen can be used by the many mitochondria in a muscle fiber for the production of ATP. Myofibrils are found within the sarcoplasm of a muscle fiber. They run the length of a muscle fiber and have a diameter of about 2 microns, and are the contractile organelle of a skeletal muscle fiber. Each myofibril is encircled by a fluid-filled system of membranous sacs called the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium ions for use during the contraction cycle of muscle. 2 important terms to note here are terminal cistern and triad. Terminal cistern refers to the dilated end sacs of the sarcoplasmic reticulum that butt up against the T-tubules from both sides, and a triad refers to the area where a T-tubule and 2 terminal cisterns are found. When stimulated, the terminal cisterns of the triads will release calcium ions that will trigger a muscle contraction. Within myofibrils, you will find smaller protein structures termed filaments or myofilaments. The myofilaments directly involved in skeletal muscle contraction are the thin or actin and thick or myosin filaments. These filaments do not extend the entire length of a muscle fiber, but are arranged in compartments called sarcomeres, which are the basic functional unit of a myofibril. Each sarcomere within a muscle cell is separated from the next via a Z-disc, which is a narrow, plate-shaped region of dense protein. Sarcomeres extend from one Z-disc to the next and contain actin and myosin filaments, which are directly involved in the contractile process. The sarcomere's organization is referenced through bands, zones, and lines. The A band refers to the entire length of the thick myosin filaments. It creates the dark band found in the middle of the sarcomere. The I band refers to the entire length of the thin or actin filament. Overall, it is lighter, less dense area of the sarcomere. You will note that the Z disc passes directly through the center of each I band. The H band or H zone refers to the area of the sarcomere that contains thick but not thin filaments. The zone of overlap refers to the area where the thick and thin filaments lie side by side. The M line is comprised of supporting proteins that hold the thick filaments together at the center of the H band. It gets its name because it is found in the middle of the sarcomere. Myofibrils are comprised of three types of proteins: contractile proteins. These generate the force during a contraction and consist of actin filaments and myosin filaments. Regulatory proteins, which function to switch a contraction on or off and consist of troponin and tropomyosin. And structural proteins. Their job is to contribute to the alignment, stability, elasticity, and extensibility of myofibrils. These include titin, alpha-actinin, myomycin, nebulin, and dystrophin. Titin connects the Z disc to the M line of sarcomeres. Alpha-actinin is a structural protein of Z discs that attaches to actin molecules of thin filaments and to titin molecules. Myomycin forms M line of the sarcomere, binds to titin molecules, and connects adjacent thick filaments to one another. Nebulin wraps around the entire length of each thin filament, anchors the thin filament to Z discs, and regulates the length of the thin filament during development. Dystrophin links thin filaments of the sarcomere to integral membrane proteins in the sarcolemma. It reinforces the sarcolemma and helps transmit the tension generated by sarcomeres to the tendons. Myosin is the main component of thick filaments. It functions as a motor protein in all three types of muscle tissue. These proteins pull cell structures to achieve movement by taking chemical energy and converting it into mechanical energy of motion, thus producing a force. A myosin molecule is shaped like two golf clubs twisted together. The handles will point towards the M line. The heads of these golf clubs, or myosin heads, contain two binding sites each, an actin binding site and an ATP binding site. The heads extend towards the thin filaments. In skeletal muscle, around 300 molecules of myosin form a single thick filament. Thin filaments are mostly composed of actin proteins. These individual actin molecules join together to form the actin filament that is twisted into a helix shape. Each actin molecule contains a myosin binding site where a myosin head can attach. Within this thin filament are two regulatory proteins, troponin and tropomyosin. In a relaxed state, the tropomyosin molecules cover the binding sites for myosin on the actin molecules, and the troponin molecules hold the tropomyosin molecules in place. It is only when calcium ions bond to the troponin that the tropomyosin molecules move away from the binding sites on the actin molecules for myosin. Muscle contraction occurs when myosin heads attach to the actin filaments and the thin filaments get pulled to the M line. You will note that as this occurs, the I band and H band or zone narrow, but the width of the A band and the length of the myosin and actin filaments remain the same. Remember that the thin filaments, or actin, are attached to Z discs, so when they are being pulled towards the M line, the Z discs come closer together and the sarcomere shortens. The shortening of the sarcomere will cause the shortening of the whole muscle fiber, leading to the shortening of the entire muscle. This leaves us with an important question to have answered. What causes the myosin filaments to attach to the actin filaments? Sarcomeres shorten through these repeated cycles, which include the myosin heads attaching to actin filaments, forming a cross bridge, a rotation of the thin filaments past the thick filaments towards the M line, and a detachment of the myosin head from the actin. In the next few slides, we will break down the cycle into 4 steps. The first step is ATP hydrolysis. The second, attachment of myosin to actin. The third step is known as the power stroke. And the fourth step, which is detachment of myosin from actin. The first step in this sliding filament mechanism is ATP hydrolysis. The myosin heads have an ATP binding site that will function as an enzyme, like ATPase, which will hydrolyze the ATP into ADP plus phosphate. This reaction will generate energy. Remember, you are breaking bonds, and that always releases energy, which will be stored in the myosin head. Once the myosin head contains this stored energy, it is energized. This stored energy causes the myosin head to assume a cocked position, which puts it into a 90-degree angle relative to the thick and thin filaments. This puts the myosin head in the proper orientation to bind to an actin molecule. Keep in mind that the ADP plus phosphate are still attached to the myosin head at this point. Step 2, the attachment of myosin to actin. After a skeletal muscle is stimulated, calcium ions are released from the sarcoplasmic reticulum. The calcium ions will bind to the regulatory protein troponin found on the actin filament. The troponin is attached to another regulatory protein called tropomyosin that is found covering the myosin binding sites on actin. The attachment of the calcium ions to the troponin causes the troponin molecule to change its shape, pulling the tropomyosin off the myosin binding sites, making them available. The myosin heads of the thick filament will now attach to the actin, forming a cross bridge. Once the myosin head attaches, the phosphate group that is stored on the myosin head is released. Note that although a single myosin molecule has a double head, only one of those heads will bind to actin at a time. Step 3, the power stroke. After the cross bridge forms, the myosin head will change its position from a 90-degree angle to a 45-degree angle relative to the thick and thin filaments. This causes the thin filament to be dragged towards the M line, or center of the sarcomere. This tension-generating event is termed a power stroke. The energy needed for the movement of the myosin head in the cross bridge comes from the energy it stored when it hydrolyzed ATP. After the myosin head pivots in this power stroke, the ADP that is stored in the myosin head is then released. Step 4, the detachment of myosin from actin. The myosin head remains attached to the actin after the power stroke occurs, until another AT molecule binds to the myosin head at its ATP binding site. Once an ATP molecule attaches the myosin head, it will detach from the actin molecule. The myosin head will then hydrolyze the ATP and will be ready again to attach to actin. This contraction cycle will continue if molecules of ATP are available for the myosin filaments and calcium ions are available for the actin filaments. The cross bridges will rotate back and forth with each power stroke, pulling the thin filaments towards the M line. One thick filament can have up to 600 cross bridges attaching and detaching 5 times per second. The force generated by these cross bridges and power strokes draws the Z discs towards one another, shortening the sarcomere. The Z discs pull on neighboring sarcomeres and the whole muscle fiber shortens. As fibers within skeletal muscle begin to shorten, they will pull on the connective tissue coverings like the endomysium, perimysium, and epimysium, as well as tendons. The coverings and tendons stretch and become taut. This tension is then transferred through the tendons, causing them to pull on the bones to which they are attached. The result is movement of a part of the body.